So Learning Unit 5 is on Chapter 8, which is on carbohydrates. So one of the themes we're going to continue to see as we learn these biological molecules is we're going to talk about first their structure and then how it applies to some functions and some applications. So like we saw with protein structure, we return to our kind of structure affords function diagram for our 4 biopolymers. So again, looking at carbohydrates, some important things to remember are the functional groups that we see in carbohydrates. So we see ketones, aldehydes, and alcohols. When we think about the linkage that we have for these, we're going to think about glycosidic bonds. And when we get to structure of carbohydrates, we'll talk about a good bit of information on kind of the anatomical details we see with carbohydrate structure. We'll talk a little bit about the ABO blood groups, potentially in sort of a take-home question. Numbers, again, that are important to think about are that they are worth 4 calories per gram, and that in terms of glucose concentration that we see in the blood, it's 5 millimolar, which translates to about 90 milligrams per deciliter. So again, that's a number that's sort of important to think about in terms of a normal blood sugar level. Again, we looked at a little bit with the structures to think about why beta-D-glucose is nature's favorite. And then we're going to talk a little bit about kind of structure in terms of understanding whether it's fuel versus fiber. So we decided that we are not going to have a fundamentals quiz. So let me cross this off here. No fundamentals quiz on carbohydrates. You are going to need to know how to draw a carbohydrate, and I'll talk about that in a little bit for your test. You will have 1 carbohydrate that you have to draw. I'm going to sort of highlight here, I'm going to circle here ones that are potentially on the table. So ribose, obviously glucose, mannose, galactose, and fructose. So even though ribulose, dihydroxyacetone, and glyceraldehyde are important, when we think about sort of the main carbohydrates that we see in terms of structure and in fuels, these are the ones that I've identified here. And particularly, it's glucose and fructose that are most important. So some of the things that I do need you to know when it comes to carbohydrate structure here is a little bit in terms of their stereochemistry. So all naturally occurring sugars are D, and that has to do with the configuration of the stereochemistry at, excuse me, I should circle this one, at this penultimate carbon. So the carbon that's next to the bottom, there's no stereochemistry in this bottom one. So you'll notice that all of these stereocenters are the same, and so that's how we get that they are D. I'm going to highlight too that there's another video on drawing structures of carbohydrates that might have some important information that would be useful to thinking about the structures of these. When we have 2 sugars that differ in the stereochemistry only at 1 carbon, we have a special name for those. We call those epimers. So epimers are 2 sugars that differ at only 1 stereocenter in terms of their configuration. So some information that's important to know is that mannose differs at carbon 2. So we say that mannose is a C2 epimer of glucose. Galactose differs from glucose at this 4th carbon right here. Every other carbon is the same. So galactose is a C4 epimer of glucose. And if you remember, right, left, right, 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 left, right, right, that tells you the positions of the OH groups on glucose, if you needed to remember. But you're not going to be asked to draw a structure of a carbohydrate in a linear form. You're going to be asked to draw it in a cyclic form. And so we're going to get to that in another slide here. So remembering cyclization chemistry is really the reaction of alcohols with carbonyl-containing functional groups like aldehydes and ketones to generate hemiacetals and hemiketals. So when we cyclize sugars, we generate what's called Haworth projections. So we use Fisher projections to represent the linear form. A number that I do want you to know is the fact that only about 1% of sugars at any one time sort of exist in their linear form, and that's because cyclization of sugars is nature's way of protecting this reactive carbonyl functional group. So we generate it as either a hemiacetal or a hemiketal, depending on whether the starting carbonyl was an aldehyde or a ketone. So by formalism, we draw Haworth projections with this anomeric carbon, that is the carbon that was once a carbonyl, on the right. And when we have kind of attack of this hydroxyl group on this carbonyl, it can attack from either the top or the bottom face, which means we have two possible configurations or stereochemistries that we can have at this anomeric carbon. And that is the origin of this Greek letter here. So this Greek letter has to do with where the positioning of this exocyclic CH2OH group is, and then the OH group that's on the anomeric carbon. So again, this is the oxygen that once was part of a carbonyl. If they are both on the same side, we would call this beta. And if they are on the opposite side, we will call that alpha. So we're going to see an alpha anomer of glucose on the next slide here. But we'd refer to this as the beta anomer. Now, something else to think about is when we do cyclize these sugars, we have the potential of using either this hydroxyl group that's on C5, which would give us a 6-membered ring, or in the case of fructose, notice that the C5 hydroxyl group is one atom closer to the carbonyl because the carbonyl is a ketone rather than an aldehyde. And so this generates a 5-membered ring, which is called a furanose sugar. So a lot of kind of anatomical detail here is provided by some nomenclature. So the Greek letter refers to the positioning of the hydroxyl group that used to be the carbonyl in relationship to the exocyclic CH2OH group. OSE reminds us that we have sugars. Furanose sugars are 5-membered rings. Pyranose sugars are 6-membered rings. When we think about the difference between where the configuration or what the configuration is on this anomeric carbon, we call these anomers. So again, this is a beta anomer. We'll see on the next slide an alpha anomer. So glucose and fructose are probably the two most important kind of fuel sugars that we're going to talk about. But what we'll be able to see here is that for glucose, we're going to see alternating groups here. And so that's going to allow us to have a unique sort of arrangement of groups when we think about the fact that this is really in a chair conformer. One other thing to highlight that I didn't necessarily mention when we saw this slide is that all sugars end in OSE, but we classify this group of sugars as aldose sugars because they have the carbonyl containing functional group as an aldehyde. All of these are ketose sugars because we have the carbonyl containing functional group as a ketone. So again, when we cyclize these sugars, it's nature's way of protecting this reactive carbonyl containing functional group. And the position of the OH group on the anomeric carbon designates this Greek letter. And this is alpha when we have this configuration, beta when we have this configuration. So glucose, specifically D-glucose here, can exist in two different anomers, the beta anomer or the alpha anomer. And to interconvert between these requires breaking this anomeric bond. So again, to get back to an open chain form, we would have to break this bond here and then recyclize coming from the opposite face of the carbonyl to generate the other anomer. So because these are isomers that result from the interconversion by bond breakage, we call them configurational isomers. So again, configurational isomers must be interconverted by bond breakage. This is different from conformational isomers where we can interconvert them by rotating bonds, and it basically just changes the spatial arrangement of groups. So again, chair flipping is what we refer to as conformational isomers. So what's really neat about beta D-glucose, so again, beta D-glucose having this hydroxyl group in this position, is we can have a conformer that puts all of the bulky substituents in equatorial positions. If we chair flip, remember when you chair flip, we have an equatorial substituent that now becomes axial. You notice that the relative position of all of these functional groups stays the same. But beta D-glucose is nature's favorite, probably because it's the only configuration that can allow for all substituents to be equatorial. And just highlighting and reviewing some things here, this is the anomeric carbon. So this is carbon 2. So if we were to switch these two positions, we would get the C2 epimer of glucose, which is mannose. If we were to switch these two groups, we would get the C4 epimer, which is galactose. So again, those are two that you need to know. So again, then we talked about additional chemistry that can happen. Once you're at either a hemiacetal or a hemiketal, you can have a second equivalent of alcohol that comes in, reacting with this carbon, which would be equating that to the anomeric carbon for our cyclized sugars. And this is how we can polymerize sugars. Alternatively, it's another way that we can add functional groups or do functional groups with other things that have hydroxyl or even amine containing functional groups. And so if we think about other possible options for these, other possible options for hydroxyl groups other than other sugars, which is how we're going to generate polysaccharides, is we have a variety of amino acids that could help. So amino acids that have hydroxyl groups include serine, threonine, and tyrosine. Amino acids that have nitrogen-containing functional groups are arginine, glutamine, lysine, asparagine, and histidine. So these 9 amino acids are what allow us to glycosylate proteins, or take proteins that have these amino acids on their surface and decorate them with sugars. So this is often how important information is displayed on cell surfaces, is you have proteins embedded in that lipid bilayer, amino acid side chains that can be decorated by the addition of sugars, which we call glycosylation. One other source of sort of a nitrogen as a nucleophile is if we think about the nitrogenous bases such as A, G, C, T, and U. And this is how we're going to add the nitrogenous base to the sugar to make nucleotides. So again, linking these guys together, if we have the hydroxyl group as another sugar, that can generate first disaccharides, but ultimately polysaccharides. So when we think about disaccharides, some anatomy that's important to consider. Again, by formalism, the anomeric carbon is always drawn on the right, but sucrose is a special kind of disaccharide where we have 2 anomeric carbons that are linked together. So this is what we refer to as a head-to-head linkage because we have the 2 anomeric carbons hooked together. Now, when you have 2 anomeric carbons hooked together, we need to have 2 Greek letters as part of the name. So again, when you have a disaccharide or a polysaccharide, the Greek letter gives you information as to the stereochemistry of the glycosidic linkage. And how we can figure that out is we can say, well, this shown in blue here used to be an OH group that was pointing down. It's on the opposite side as the exocyclic CH2OH group, so this guy is alpha. If we look at this anomeric carbon for fructose, and again, fructose is going to have the anomeric carbon at carbon 2 because it was a ketose sugar. This used to be a ketone. And if we look at the position of the OH group, which is shown here in green in relationship to the CH2OH group, it's on the same side. So the configuration of that anomeric carbon is beta. So when we're giving information about the linkage of disaccharides or polysaccharides, we not only need to indicate the configuration at the anomeric carbon by a Greek letter, but we also need to give information as to which carbons are hooked together. So the glycosidic bond in sucrose would be labeled alpha beta 1, 2. And understanding the nomenclature of this linkage is really important because any linkages that we see are alpha, we're going to label as fuel. This is again a unique sugar because both of the anomeric carbons are part of the glycosidic bond, so we have this head-to-head linkage. We refer to this kind of sugar as a non-reducing sugar. There is not a free head group. There's not a free anomeric carbon that's part of a glycosidic bond. Or I should say there's not a free anomeric carbon. There's no anomeric carbon that's not already part of a glycosidic bond. So thinking a little bit more just about some things that we talked about with sucrose here. With our disaccharides and polysaccharides, there's 5 that I need you to know. I do want you to know who the components are. So sucrose is made of glucose and fructose. I want you to know the linkage. So the linkage in sucrose is alpha beta 1, 2. And again, these small numbers here remind us that it's a head-to-head linkage. Just another reminder here that this is condensation chemistry when we link these sugars together and we eliminate a small molecule of water. This will become important because when we study enzyme chemistry in a couple of chapters, we're going to talk about using hydrolysis chemistry to break these glycosidic bonds. And actually all of the linkages that we talk about, whether it's a peptide bond linkage, a glycosidic bond linkage, or an ester linkage, we tend to see hydrolysis chemistry to facilitate that. When we think about lactose, again, lactose is what's found in milk sugar. Lactose is a disaccharide of galactose and glucose. When we think about the carbon that's present as part of the glycosidic bond, it's actually the anomeric carbon from galactose. You don't need to know that, but I do want you to know that the linkage is a 1,4 linkage in the beta configuration. So we would say that it's a beta 1,4 glycosidic linkage in lactose. The fact that it's this beta linkage means we're going to call it fiber. Now, that's not to say that lactose is fiber, but this beta reminds us that this is not something that tends to be easily digestible or normally digestible in our body. We talked about the fact that we as mammals are unusual as humans in that we consume lactose and milk sugar post-weaning. Most animals, once they kind of are weaned, they lose the ability to produce lactase, the enzyme which is used to digest and break the glycosidic linkage in lactose. And so in losing that enzyme, they lose the ability to digest lactose and they become lactose intolerant. So one of the things that we want to highlight that's different about lactose compared to sucrose, because it's what we call a head-to-tail linkage, we do have an anomeric carbon that is free, that is not part of a glycosidic bond. And so having this free anomeric carbon, we call this a reducing sugar, meaning it's one step away from having a reactive carbonyl-containing functional group. All we need to do is break this bond for that. To have either one of these carbons be a carbonyl-containing functional group again, we'd not only need to break the glycosidic linkage, but then we'd need to also break these anomeric bonds to linearize the sugar. So the difference between reducing and non-reducing ends is going to become very important as we kind of delve into the details of how glycogen is metabolized in your body. We're not going to get into any details with this until Biochem 2, but one of the things that we want to think about is the more non-reducing ends we have, the more reducing ends that we have in our glycogen polymer, the faster we're going to mobilize glucose. So that'll become important as we look at some of the important polysaccharides and consider the concept of branching. So polysaccharides that we talked about, obviously the most common one to think about is just a homopolymer of glucose. So what's interesting is we can have a homopolymer of glucose where we have beta 1,4 linkages. This is what happens when we have cellulose. So cellulose is a homopolymer of glucose linked by beta 1,4 glycosidic bonds. And again, we can see that this is a beta linkage because this was an OH group that was pointed up. On the same side is this CH2OH group, and so that is going to generate a beta linkage. We know that cellulose is not digestible by us. This is really what makes fiber in our body. I'm going to contrast that by coming down to something called amylose or alpha amylose. That alpha again reminds us that it's an alpha 1,4 linkage that we have between these glucose residues. So alpha amylose is a homopolymer of glucose linked by alpha 1,4 glycosidic bonds. So this makes this a fuel in your body. This is what's prevalent in potatoes. This is what's prevalent in apples. So again, lots of starch that we have in alpha amylose, lots of cellulose or fiber that we have in apples. I'm going to move up to the top here and talk about a variant of alpha amylose, which is a branched version of plant starch. So if we imagine taking this linear homopolymer of glucose with alpha 1,4 linkages, and we take this CH2OH group, this 6 carbon, and we use that now as an OH group to create a glycosidic linkage between an anomeric carbon, we create what's called a branch point. And again, we're going to give some nomenclature to this and call it an alpha 1,6 branch point. So amylopectin is essentially a branched version of alpha amylose. Both serve important features and functions in plants, but again, we're going to see the importance of branching when we understand glycogen metabolism in our bodies. The last one is one that I don't need you to know the structure of. I do want you to know a little bit about chitin. So chitin uses an N-acetyl modified glucose, and it polymerizes this using a beta 1,4 linkage. So chitin is what's present in the exoskeletons of bugs and crustaceans, and you cannot digest that, again, because it's a beta linkage. So we talked a little bit about glycogen, again reminding you we can refer to glycogen as animal starch. So starch, meaning it's got alpha 1,4 linkages with alpha 1,6 branch points, So this is your equivalent of sort of a storage form of glucose in your body. You store it in two main areas. You can store it in your liver, and you can store it in your muscles. So about 5 to 6% of the weight of your liver is actually glycogen, and about 1 to 2% of the weight of your muscles is actually glycogen. So what's really interesting is what this glycogen is able to do. Now, in both cases, it's able to be broken down into free glucose. But in your muscles, it's meant to be used locally. Your muscles are kind of selfish in that regard, that they have this reserve of sugar, but they're just going to break it down and use it for themselves. This is in contrast to the glucose that's broken down by the glycogen polymer that's in your liver. When that glucose is broken down, it's released to the bloodstream and becomes available for any cell to use. So we say that that's available for global use. So how does this glycogen get released and used by the body? Well, there's two main hormonal signals that we're going to talk about in more detail later, but introducing them now. One is secreted by your pancreas, and the pancreas secretes glucagon in response to low blood sugars. So glucagon then is only going to be a hormone that is sensitive to the liver, because the liver is the only one who's going to allow its glycogen to be broken down to glucose for global use. Muscle cells have no interest in responding to a call for low blood sugar. So they have no receptors for and thus do not respond to the pancreas's release of glucagon. Your adrenal glands, in kind of a fight or flight response, produce and release epinephrine. So what's interesting is epinephrine can actually respond or have responsiveness in both of these tissues. So again, an epinephrine response in the liver will do the same thing as glucagon. It will depolymerize glycogen, break those glycosidic bonds, generate free glucose, release it to the bloodstream, and allow it to be used by any cell that might need to respond to this fight or flight situation. Epinephrine also will respond, or is responsive in muscle cells. And that's because if you need to fight or flee, your muscles need to have sufficient energy so that they can use it locally. So just a reminder, we want to be thinking about when we talk about some of these hormones, when we talk about some of these extracellular signals, what are they? Are they need state or fed state? And again, when we talk about need state, we're talking about the need for energy or the need for remedying a low blood sugar level or a high blood sugar level for that case. But in this case, this need state is a need for energy or glucose. So I do want you to continue to remember that need state signals put us in a phosphosystem, which means we are going to be decorating things with phosphates via kinase enzymes. So again, the flip side of that is if we're in a fed state, we're going to be in a dephosphosystem, and we're going to be using phosphatases to kind of remove those phosphate decorations. The last sort of big picture thing that we talked about in this chapter, and this was a fairly busy slide here, so I'll try to go through it slowly, is to talk about the difference between an insulin response and insulin resistance. So we've got this figure sort of numbered here. This part that's down at the bottom here where we're talking about glucagon, that's actually what we refer to down here. So glucagon is your body's response to low blood sugar. So what we're going to be talking about up top here is what happens if you're in a fed state. That is, you have an elevated level of blood sugar. So we're going to go around here. We've got these numbered, so we'll kind of start over here with number 1. When you have an increased concentration of blood sugar, the response is that your pancreas secretes insulin. And just as a reminder, if you have type 1 diabetes, that means your pancreas cannot produce insulin. So you actually don't even get past this first step. People who are type 1 diabetics need to take insulin in the form of a shot, or if they have an insulin pump, to provide that exogenously because they're not able to produce it endogenously. But let's assume that you're not someone who has type 1 diabetes. Elevated blood sugar is going to result in an increase in insulin, and we're going to talk about 3 main tissues that sort of respond to this signal. We're going to kind of come up here first, and we're going to talk about the muscle's response to an insulin signal. When your muscles get a signal that there is high blood sugar in the form of that insulin, receptors on the cell surface bind to insulin and they circulate a transporter called GLUT4 to the cell surface. So this is really kind of interesting because GLUT4 isn't normally on the surface of your muscle cells. It's only put on the surface of your muscle cells when there's a signal like insulin to bring it to the surface. Once they have that surface receptor there, they're going to bring glucose in and they're going to polymerize it into glycogen. A similar mechanism happens in your liver, but I'm going to highlight here that it's a different transporter. We'll talk more about these transporters in our chapter on membrane transport, but GLUT2 is different not only in its function, which we'll delve a little bit more deeply into, but in the fact that it's always present on the liver surface. So in always being present on the liver surface, it doesn't necessarily mean that there needs to be this insulin signal. As soon as glucose levels rise, the liver will bring in glucose and it will also polymerize it to glycogen. So again, both of these tissues will replenish this short-term reserve. I do want you to know that glycogen will last for about 12 hours in your body. It's not to say that all of a sudden at 12 hours, like it shuts off. It starts slowing down prior to that, but there's about 12 hours of steady kind of supply of glucose from the glycogen stores you have in your body. So once you replenish these stores, any glucose that's excess will be converted in your liver to triglycerides. And we'll learn about triglycerides a little bit more in our next chapter, but if you remember that they're worth 9 calories per gram, this is nature's most efficient way to store energy because it's worth so much more. So any excess carbohydrate, actually any excess fuel at all, is going to be converted into triglycerides in your liver, and then they're transported to your adipose tissue for storage. So one of the important implications is insulin's ability to kind of respond and tell adipose tissue what to do. So when adipose tissue senses insulin around, it activates fat storage. So it sort of opens up its transporters to bring in fatty acids, bring in triglycerides, and store them as fat. And it also inhibits fat breakdown. Kind of these two systems together are opposite of one another. And we'd have what we call a futile cycle if we had fat storage operating at the same time as fat breakdown. So these opposing pathways metabolically, we will have lots of examples of these in our body, won't operate at the same time because then we'd just be literally spinning in a circle. We'd be storing something only to break it down, storing it, breaking down. Your body's not inefficient like that. So it's very normal when you're in a fed state to activate storage and then inhibit breakdown. If you're in a fed state, you don't need to be breaking anything down. But again, herein is part of the problem when you have high levels of insulin sort of chronically, is you're not able to tap into any reserves that you have, and unfortunately that means you can't lose any weight that might be stored. So that's sort of the normal insulin response. So let's talk about what happens with insulin resistance. So with insulin resistance, we really sort of put earmuffs on at this point. Blood sugar levels rise, your pancreas produces insulin, you've got plenty of insulin around, but these cells that are normally, these tissues that are normally responsive to that signal are not listening. They've got earmuffs. So what that means is your muscle cells are no longer going to be taking up that glucose. Your liver cell is going to be impaired in its ability to take in that glucose. And what that really means is you're going to have still high levels of insulin and sugar in your bloodstream, but these cells are going to be starving because they do actually need the glucose, but they've lost the ability to become, to listen to that signal. They've become resistant. It's almost like the boy who cried wolf. They're no longer listening to that signal. And at this point, the only pathway, the only option that's still kind of active is this one. Your body kind of taking and storing that excess glucose as triglycerides in adipose tissue. One of the big picture things to sort of highlight is to remember that elevated blood sugar is really a toxic situation in the long term. Glucose levels are meant to rise and fall and rise and fall, and insulin's supposed to be the signal that allows them to fall. But again, if you're not able to listen to that signal and these pathways are no longer operational, your body needs to lower its blood sugar levels, and it's desperately trying to do so by throwing any excess sugar into adipose tissue. And so unfortunately, one of the situations that you see is people who develop this type 1 diabetes, the hallmark of type 2 diabetes, is that you're not able to listen to this insulin signal, is that you tend to put on weight. And you tend to be hungry all the time because your cells are starving, but you're throwing all of the food that you're putting into your body into fat storage. And so really, really tough situation to be in. So a few solutions that we're going to talk about very briefly here. A little bit more information will be kind of delved into potentially in a take-home question, is how a three-prong approach can be used to help treat type 2 diabetes. So from a dietary standpoint, by reducing or eliminating carbohydrate, by having a low carbohydrate diet, you're basically reducing sort of this initial stimulus so that you hopefully reduce levels of insulin and allow insulin resistance to sort of go away. You actually become more sensitive. You have insulin sensitivity. Exercise is another sort of prong of this approach to treating type 2 diabetes. And what that does is it actually helps increase what we see over here. Again, if the muscles were listening to the signal, they would have put GLUT4 on the cell surface, which would allow the input of glucose into those cells. Exercise is a way to sort of force this process to happen, even if the signal for insulin isn't being adhered to. And lastly, prescription drugs are possible. So one that we're going to talk about briefly is metformin. And metformin, we're not going to talk about the mechanism of action or anything right now. We'll save that for another learning unit. But metformin increases insulin sensitivity, and importantly, it inhibits gluconeogenesis. We're not going to get into the details of how this happens, but I do want you to know this term. Gluconeogenesis is how we make carbohydrates from scratch. Remember that you do need to have a homeostatic level of blood sugar all the time in your body. And so if you haven't been consuming a lot of carbohydrates, if maybe you've been doing a lot of exercise, your body can make the carbohydrates it needs from scratch. And again, blood glucose levels are important because there are some cells, like we've learned about our red blood cells and hemoglobin. Red blood cells subsist exclusively on glucose as a fuel. We'll delve more into that when we study our chapter on glucose metabolism and we understand what kind of happens in glycolysis. So that's it for our learning unit on carbohydrates, specifically for what you need to know for the in-class portion of the test. There's going to be a lot of stuff that we'll talk about for the take-home version of this test. Importantly, we'll talk about things like artificial sweeteners, we'll talk about alternative sweeteners, and we'll talk about, lots of things like glycemic index and things as they relate nutritionally to the consumption of carbohydrates.